Okay, so I want to start with a reminder of why cells divide. And so hopefully you can see my whiteboard. Of course, you know, I finally get the iPad and the Apple Pencil, and it's not working today. It's saying that I don't have the Apple Pencil plugged in when I do. And anyway, so here we are, back to the board. So just a reminder of why cells divide to start with. Okay, first reason cells divide is growth. So for you to get to adult size you, a lot of cell division had to take place. A whole lot. From going from just one cell to trillions of cells took a lot of cell division. And development is really in that same category. So development would be going from that single cell to a multicellular organism would be development. Growth is really just getting bigger. So really, I guess development could go before growth. Repair and replacement of damaged cells. So if you've watched the videos, you know that certain cells can be repaired and replaced and certain cells cannot. So for example, muscle cells can be repaired and replaced, but once you reach adult size, the cells of your brain and spinal cord cannot be repaired or replaced. So these are kind of two different types of cell division. Some of your cells divide all the time. Obviously, if you cut yourself, your skin cells are going to divide and repair, replace those cells. If you work out your muscle cells really hard through an intense weight workout, you're damaging those cells and you're stimulating them to divide and repair themselves. But then there are certain types of cells that can never divide again. Once those cells reach maturity, they can never divide again. And that's unfortunate. And a lot of brain and spinal cord research focuses on how do you re-stimulate the cell cycle to start again for those cells? Because at some point they divided to get to adult you. I mean, your brain didn't start out the size it is now. So how can we turn back the clock on those cells and stimulate them to start dividing again and repair that damage? And we haven't figured that out yet. That's a big deal in science. Okay, and then the fourth reason that cells divide is to produce gametes. And that's a different type of cell division. So all of this type of cell division, this is all what we call mitosis. Division of what we call the somatic cells. Remember, the somatic cells are every cell of your body except eggs or sperm. So growth, development, repair, replacement, that all happens through mitosis. And in mitosis, you're producing two genetically identical cells. And that's very important to understand because you want all of your cells of the body coding for proteins with the exact same recipe. You don't want a whole bunch of different codes for how to make a protein. You want it all consistent. So mitosis has the goal of producing two identical cells every time. The last reason that cells divide, making gametes, it's a different type of cell division and the goal is different. So the fourth reason why cells divide is to produce gametes. And all sexually reproductive organisms produce gametes, eggs and sperm. Some organisms can produce both, and some just produce one or the other. You might not know that even plants have eggs and sperm. And the sperm has to get to the egg and fertilize it to produce a new plant. When we produce gametes, that type of cell division is called meiosis. And the goal is not to produce genetically identical cells. In fact, the goal is the complete opposite. The goal is to reduce the number of chromosomes to half and to produce genetically different gametes. So goal of meiosis, very different than goal of mitosis. And I'm going to summarize that in a minute. But those are the reasons that cells divide. And it's just really important to understand that when we're producing gametes, it is a different type of cell division than the whole rest of the body. Any questions about that part? Okay, and then just quickly to review the goals of mitosis and meiosis. The goal of mitosis is to produce two genetically identical cells. And we start with a cell that has the full number of chromosomes. And remember, we call that a diploid cell. It has the double number of chromosomes, which for humans is 46. But this is true of all organisms. Okay, in mitosis, they start with a cell that has the double number of chromosomes. And at the end of mitosis, you're going to end up with two genetically identical cells that also still have the double number of chromosomes. So the chromosome number stays the same, and they're genetically identical to each other. Meiosis, on the other hand, is different. In meiosis, the goal is to produce, sorry, this pen is terrible. The goal of meiosis is to produce 4 genetically different cells. And we do that through 2 rounds of division. That's how we end up with 4. So we start off, we make 2, and then we divide again and make 2 more. So we're going to produce 4. They're going to be genetically different rather than genetically identical. And you start with a diploid cell, but you end up with 4 haploid cells, half the number of chromosomes. So I don't know if you can see the bottom of the board really fast, but we're going to start off with a 2N cell and go through 2 rounds of division, and we're going to end up with 4 cells that are all haploid, half the number of chromosomes. Okay, so in humans, that means 23. So in humans, what we call a diploid cell, which has the double number of chromosomes, that's 46 chromosomes. And the haploid, the N number is 23. So this is what our eggs and sperm have, is 23. Okay, and the reason you have to go through that reduction division to make gametes is otherwise every generation you would double the number of chromosomes. So if you have an egg with 23 and a sperm with 23, that gives you that initial cell of 46, which is called a zygote. And that now goes through mitosis to produce every cell of the body, including the eggs and sperm. Okay, so your eggs and sperm had to come from one of these cells with 46, and it needs to get back to 23 again. Any questions about that? Okay, so the purpose of my talk today is to talk a little bit about what can go wrong. What can go wrong in mitosis? What can go wrong in meiosis? Why one is worse than the other? Okay, an error in meiosis is far worse than an error in mitosis. And also to talk about cancer, because cancer is an error in the cell cycle. It's cells that start dividing out of control because some type of cell cycle regulator has gone awry. And at some point, we will all be affected by cancer in some way. It is the one medical problem we still don't really have a handle on. That and the coronavirus, apparently. But any questions at all just about the basics of meiosis and mitosis and what happens at each stage, what the goal is of each one? The lab this week does a really good job of reviewing that. Okay. So let's just real quickly talk about errors in cell division. Okay, my first example, I'm going to have a skin cell. And it's time for this skin cell to divide, either because it's just lived its life. So most of your skin cells live about 30 days. Every 30 days, your skin cells divide and completely replace themselves. Or maybe you got cut, so these skin cells need to repair and replace themselves. Okay, here's your next question for attendance purposes. Is this skin cell going to go through mitosis or meiosis? Yeah, so this skin cell is what we call a somatic cell, which again means every cell of the body except for eggs and sperm, and it's going to go through mitosis, which means it's going to produce two genetically identical cells. But let's say it doesn't. Okay, so remember what's going to happen in mitosis. I'm going to draw a cell. So remember, in prophase of mitosis, the chromosomes condense and become visible. The nuclear envelope breaks down and disappears, and we end up with chromosomes that are now visible. And I'm just going to draw two pairs of chromosomes. And as you know, if you've seen a picture of a karyotype, not all of our chromosomes are the same size. Now, the goal of mitosis is to pull those two sister chromatids apart. Remember, we made this second sister chromatid in the S phase of the cell cycle. And now those are going to attach to microtubules from opposite poles of the cell. And the goal is to pull those sister chromatids apart. And you learned all about how that happened in the lecture videos. But let's say that something goes wrong and this one doesn't correctly attach to a microtubule. And now what's going to happen is this cell, when this divides, this cell is going to end up with too much DNA and this one won't have enough DNA. Okay, so when this thing divides, this entire chromosome is going to go to this half of the cell and nothing's going to go to this half. So we're going to end up with two skin cells that have the wrong number of chromosomes. One is going to have too many and one isn't going to have enough. Okay, so the end result of this, if we look at all 46 chromosomes for humans, is we're going to end up with one cell that has one too many, 47, and one cell that has one too many, not enough, 45. So this is missing one because both sister chromatids went to this one. So there's too much DNA in this cell and not enough in this cell. Okay, and remember, these are skin cells. Is this catastrophic? Yes or no? Is this a big deal if this happens? No, this is not a big deal. Why? Because remember that clock of the cell cycle, which is really important today. G1, S, G2, M. And remember, we have checkpoints. We have that G1 checkpoint here that's really the most important one. And what will happen is after these cells divide, so we have the cell with 47 and we have the cell with 45, and they're starting their new little baby cell life, they're growing, and at some point they're going to go through this checkpoint, and that checkpoint will hopefully, if everything's working right, catch the fact that this one has too much DNA and this one doesn't have enough, and those cells will be destroyed. Those cells will be taken out of the cell cycle, so they're not allowed to replicate their DNA and divide again, making more bad cells. So an error in mitosis is not a big deal. We have cell cycle checkpoints that will destroy those cells and not allow them to divide again. Also not a big deal because you have thousands and thousands and thousands of skin cells, right? Like, big deal. This is 2 skin cells. And if you go spend just a few minutes in the sun, you're going to get some DNA mutations in your skin cells. No big deal. The cell cycle checkpoints take those cells out of the cell cycle, so bad cells are not allowed to divide. Any questions about that? Okay, then let's compare that with an error in meiosis. And errors in meiosis can happen in eggs or in sperm. As you probably already know, errors in meiosis become more common in females in making eggs with age. So there is a strong correlation between eggs and errors in meiosis in females. So that's because we're born with all of our eggs already through meiosis 1. And then every 30 days, an egg goes through meiosis 2. So by the time you're in your 40s, those chromosomes have been sitting there for a long time at meiosis 1, and the chances of error in pulling those apart and having everything connect correctly, it's just really unlikely that things will go well. We don't even know the rate at which it happens because only a few of those mistakes produce a baby, and so we don't even have statistics on how often that happens. But errors can also occur in sperm production, and those are not related to age. Those are related to just random mistakes that happen, because random mistakes can happen. It doesn't always go perfectly every time. It's also influenced by things like smoking marijuana and taking other drugs that results in errors in meiosis when sperm are produced. So let's say we have a cell with 23, and we're going to say that this is a sperm cell, because, you know, eggs always get a bad rap. That we know a lot about the disorders that result from a bad egg, or an egg with the wrong number of chromosomes. So we're going to say that the sperm has the wrong number today. But we start with a normal sperm. Oh, I'm sorry. I'm saying this the wrong way. This is a precursor to a sperm, okay? This is a cell that's going to become a sperm, and you don't need to know the origin of this cell. It just came from a germline cell. This is called spermatogenesis. You'll learn about this in anatomy, but this is a cell with 46. It's just a normal germline cell, same 46 chromosomes that you have in every cell of your whole body. And it goes through the 2 stages of meiosis, and something didn't happen correctly, just like I showed you in the mitosis picture, where maybe a microtubule didn't connect correctly, or maybe the microtubule connected, but for whatever reason, it didn't separate those chromosomes. Now, remember, in meiosis, that can happen at either stage. So in meiosis 1, the homologous chromosomes separate, which means the 2 number 1s, the 2 number 2s, the 2 number 3s, those separate intact in meiosis 1. So something could go wrong there. Maybe the whole homologous pair stays together, and then you really have not enough and too much DNA in one of the cells. Okay, or maybe the mistake happened in meiosis 2. It can happen either way. And this is when sister chromatids separate. Sorry, that says chromatids over there. I can't get my arm over there to write. So that mistake can happen in either one of these, but now we end up with, you remember, the goal was to produce 2 cells of 23 and then 2 more cells of 23. So we were going to end up with 4 cells of 23, but now we're going to end up with 2 that have 24 and 2 that have 22. Remember, normal is 23. That one set of these ended up with too much DNA, and then another set ended up with not enough DNA. Now let's say that all of these sperm go on to fertilize normal eggs. So let's look at what that looks like. We're going to do one of the 24s and one of the 22s. Okay, so normal egg with 23 and another normal egg with 23. And now we have a sperm with 24 and a sperm with 22. Obviously, if we do the math on that, this zygote is going to have 47, and this zygote's only going to have 45. Okay, this is catastrophic, because now what's going to happen is that first original cell, this zygote, is going to divide, and now every cell of both of these organisms, whether this is a frog, a sunflower, a mushroom, or a human, no matter what it is, every cell of this entire organism now has the wrong number of chromosomes. And there's no way to fix that. Okay, this is completely different from 2 skin cells having the wrong number of chromosomes, and they get kicked out of the cell cycle and no big deal. This is now going to result in an organism that has the wrong number of chromosomes in every single cell. So this organism would have too many, and this one would not have enough. And remember, I talk about in the videos, and also there is, a part of the lab this week that talks about what is viable and what is not viable in a human. So there are a few cases where you can have 47 and it will result in a baby, but only one of those can live to adult age in the autosomes. Okay, so remember those 2 terms, autosome and sex chromosome, are an important part of this story too, because it really depends on which chromosome you have too many of or which one is missing. So if you're missing one, the only one you can be missing and still develop is an X. You can't be missing one of your autosomes. So just to review that from the videos, there are no viable autosomal monosomies. So So monosomy, that means one body. That means you just have one of that chromosome. Gosh darn it. I'm so sorry. I keep not realizing you can't see the whole board. So there are no viable autosomal monosomies. That means for chromosomes 1 through 22, that's the autosomal part, you can't just have one. That baby never develops. There's only one viable sex chromosome monosomy, and that is just having one X, and it's called X0 because you have one X and you don't have the other chromosome, and that's called Turner's syndrome. So the only way this one with 45 would actually develop is if the chromosome that's missing is the X. Otherwise, that one with 45 would never develop. Any questions about that? So this one that's got 47, that means that baby is going to have a trisomy, which means 3 of a chromosome. So 2 is the normal number. If you have an extra one, it's called a trisomy. So there are 3 viable autosomal trisomies. (13, 18, and 21. And remember from the lecture videos that it's really related to the number of proteins that those chromosomes code for and what proteins those really are. What that means, so remember autosomal means chromosomes 1 through 22. That means the other 19 chromosomes have too many critical proteins. And that baby never develops because those are too critical to survival. And you're really not making normal versions of those proteins if you have an extra chromosome. 21 just so happens to be the smallest, and it codes for proteins that aren't critical to survival. And that is a whole range of ways that can manifest itself. So remember chromosome 21, that causes Down syndrome when you have a trisomy. And that is the only one that can result in an adult human. 13 and 18 usually die in utero. Sometimes are a live birth. If they are a live birth, those babies usually don't live beyond a year. And if they live to a year, it's only with extreme medical intervention. There are sex chromosome trisomies. And those are talked about in the lab exercise and also in the videos. So you could be XXX, you could be XXY, you can be XYY. So there are trisomies of the sex chromosomes. And for the most part, you wouldn't know looking around the room if a person had a sex chromosome trisomy or not.
Professor, can I ask a question?
Sure.
I just was looking at the lab before class, and I saw that you asked a question asking why you can't have a trisomy with other autosomes. I just was wondering if you wouldn't mind touching on that a little bit.
Yeah, that's exactly what I was saying for this one. That's a great question. So if out of 22 chromosomes, we can only have 3 different ones that even result in a baby developing, that means for those other 19 autosomes, they just code for too many critical proteins. And if you just have one of a protein, or if you have if you have one of a chromosome or 3 of a chromosome, you don't make the protein correctly. So to make a correct version of a protein, you must have 2 copies of that gene. So in other words, on 2 chromosomes. So if you have just 1 or 3, you can't make that protein correctly. So you guys already know, a bit about how we make proteins. You're about to learn even more about how we make proteins. So remember, the DNA codes for the messenger RNA, and then the messenger RNA takes that code out to the ribosome, and it tells the ribosome how to assemble that protein. Put this amino acid first, this one second, this one third. To do that correctly, you need 2 different copies of that gene. If you have just 1 copy, or if you have 3, you can't make that protein correctly. So then the question becomes, okay, what does that chromosome code for? And if it codes for proteins that are critical to survival, that you can't live without, then you're not even going to develop. Okay, so if it's the protein that makes your heart develop, or makes your brain develop, or makes your liver develop, or a critical enzyme that you can't live without, an enzyme that carries out cell respiration, you can't function without that protein, and so that baby is never going to develop. The 3 trisomies that do develop, it's because the proteins that those chromosomes happen to code for, you'll have some serious problems, and that's why 13 and 18 don't live. But even like with trisomy 21, those individuals have not only problems with mental development, but physically have a lot of different issues. It's just that particular chromosome doesn't code for proteins that are critical to survival. So it's not any critical enzymes that you can't live without, those kinds of things. And it is the smallest little chromosome too. It doesn't code for nearly as many traits. That's a great question. Anybody else with questions about that? And, most of us have known someone, or, at least had some experience with people with Down syndrome. And it really is a full spectrum of disability and ability. My husband's cousin, Cindy, just died a few months ago. And she made it to age 60. And she lived an independent life. Until the last 2 years, where she really started having some serious medical issues. But she had a boyfriend her whole adult life. She had a job. She took the bus by herself every day to her job. So
that's awesome.
Yeah. Yeah, she was a very cool person. Funny.
Yeah, I feel like everyone who, any person who has Down syndrome can be so different to the next one and their abilities.
Absolutely. And then, that brings up the whole heredity versus environment question too, and how those individuals are treated and raised from the time they're little. And I just know in Cindy's case, she was just a member of the family from day one, and she was never treated any differently. They had the same expectations of her in terms of household chores and contributing to the family, communicating, all of that. So there are a lot of studies about how much of a difference that makes, but sometimes it's just that person's medical condition and mental condition doesn't allow them to lead a normal life, no matter how much nurturing and love and all of that they get. So we're still trying to understand why that happens. Because as you guys know, your DNA is only part of the story always with all disorders. And we're going to talk about that today with cancer. Your DNA is only part of the story. And then other things influence how your DNA gets expressed. That's kind of the interesting part of the story. Okay. So if there's an essay question about what is worse, an error in meiosis or an error in mitosis, if any of you say mitosis, which typically about 80% of people on a test still say mitosis. If any of you say that, I'm driving over to your house and I don't know, I'm going to just have another conversation with you in person, socially distanced or something, because it's really, really, really important to understand why those are different. Really important to understand why they're different. And hopefully, you know, after the cancer talk today, you'll understand that there are things that can go wrong with somatic cells too that are devastating and cause people to die. But a normal person with an error in mitosis, it's not a big deal. Meiosis, again, that is a really big deal. Every gene on that chromosome that's either missing or extra is affected. And it's just that entire individual has the wrong number of chromosomes. Now we have technology today where we can fix that. So with CRISPR, and I'm going to talk about CRISPR a lot next week because we're going to be talking about how genetic disorders arise, how they're inherited, because now you know how eggs and sperm are produced, and we can start talking about how genetic disorders arise. And you're going to actually be making a discussion post in 2 weeks about genetic disorders. But CRISPR is a new technology that allows us to go into an embryo and correct that mistake. And it's a big deal in science today. Ethically, you know, is that okay? Should we be messing with the human genome? Obviously, it can't happen in utero, so we're talking about in vitro fertilization. But if you have an embryo that has a genetic disorder or it has the wrong number of chromosomes, we have the ability to fix that now. We're doing it in mice and other organisms. And in China, they're actually doing it in humans. But in the US, ethically, we have decided not to do it with humans yet. But we do have the ability. The technology is there. Okay, so let's talk about cancer and what goes wrong with cancer. For that, I'm going to do a screen share. Because my iPad's not working and I just have too much to tell you to just keep erasing this board, I really wish my iPad was working. I threw this together in about 30 minutes today, so it's not the best, but it's the best I could do considering it was a last minute thing that I had to do. Okay, so I just want to talk to you a little bit about what causes cancer, what is cancer, and give you some kind of current statistics in the US on cancer. So I'm sorry, I don't normally like to have this many words on a PowerPoint, as you know. I'm really against that, but today I'm so sorry, I just didn't have another choice. So you might have to, like I'm doing right now, you might have to move your view around so that the pictures of people are not affecting your view of the slides. So this is pretty wordy, so please don't copy this exactly. Wait, let me see the chat here. Can you guys see my screen?
Yeah.
Okay, great. Okay, so really what happens in cancer is, for different reasons that we'll discuss, it takes one cell to go bad. Okay, one cell that starts dividing in an unrestrained manner and eventually invades and damages surrounding tissue. And if things get really bad, those cells can break off and travel to other areas of the body and damage that tissue also. And we're going to talk about why you can't just take out that tumor and restore normal function to that organ. We'll talk about that today too. Sorry, I'm trying to get this moved around to where I can make this work. Okay, so the reason that cancer cells behave in this abnormal way is there are changes in their DNA. And so we call not every gene causes cancer. Okay, so you can have a mistake in a gene and it can cause a certain genetic disorder, or it can cause that protein to not be made correctly, but it's not going to cause cancer. It's just certain genes that when they have a mistake will cause cancer. And those genes really fall in two different categories that we're going to talk about. And you'll understand this a little better now because you know a little bit about the cell cycle and those checkpoints that happen. Okay, so this is kind of just the quick stats. I'm going to give you some more detailed stats in a minute, but 1 in 3 people in the Western world develop cancer and 1 in 5 die from the disease. So that's a big deal. In my own life, my mom died of cancer when she was 50. My youngest sister was 11 when my mom died. She left behind 6 kids. She raised us all herself, and it dramatically changed our lives. I mean, the people we are today, it's a result of our mom dying young from cancer. And now, over the past 2 years, my stepson has had cancer and has been going through treatment. And I'll tell you, the difference in treatment now versus 23 years ago when my mom had cancer, it's astronomical how much progress we've made. I mean, even in the past 2 years, with my stepson's leukemia, he has a specific gene mutation that is causing his leukemia. And they said that even a year prior, or maybe even 6 months prior, he would have died. And he is now 100% in remission, cancer-free, because of the drugs we have today that we have developed even just that recently. He was part of a clinical trial for a brand new drug that had only been on the market in clinical trial for 2 weeks when he was diagnosed. So timing is everything. You know, the same thing happened with HIV. Not many people in the US today die from HIV because we've made so much progress on the drugs that are available. And so many people died just short of having those drugs developed. And it's so tragic because you think, well, gosh, if they had contracted it today, they would still be here. And really the same goes for cancer. You know, there are a lot of cancers where even 6 months ago, people were dying who we can save today. So that's good, right? That's the kind of progress we want to keep making. If we've ever been through that before, we know we don't want anyone else to have to go through that. And I could go on and on about why more cancer research doesn't get done in the United States, but I'll save that for another time. There are currently about 200 types of cancer that we know of, each with different causes and symptoms and treatments. And your risk of developing cancer really depends on a lot of factors. And it kind of has to be the perfect storm. It has to be the genetic mistake, and then you exposing yourself either unintentionally or intentionally to different factors that increase your risk. Obviously, age plays a big factor. As we get older, we're just more prone to DNA mistakes, and we start losing the ability to fix those mistakes. And then obviously your DNA plays a role, and your lifestyle choices play a role. There are really kind of 2 different kinds of mutations that cause cancer. When they say germline mutation, what they're talking about is you got this mutation from the egg or sperm that made you. In other words, you inherited that mutation from one of your parents. And it's now in every cell of your whole body. So just like we were just saying, you know, getting the wrong number of chromosomes can be a problem. Inheriting a mistake in your DNA from one of your parents can also be a huge problem. And one of the things you can inherit from your parents is a bad gene that can cause you to get cancer. But believe it or not, most cancer is not caused by inherited mutations. It's caused by what are called somatic mutations, and these are changes in your DNA that happen during your lifetime. So things you're exposed to during your lifetime. Again, either intentionally or unintentionally. Stress, pesticides, plastics, pollution, secondhand cigarette smoke, other contaminants in your food and drinking water. Lots of things that we're exposed to that we don't even realize are causing these changes in our DNA. So this is just really kind of explaining that a little bit better. So somatically or inherited. So you can either inherit it from your parents, in which case it's present in every cell of your body. And really, I'm going to talk to you in a minute about what those kinds of mutations are, but these are primarily in those tumor suppressor genes. So remember, if you've watched the video on the cell cycle, one of the biggest things that can go wrong that causes cancer is that bad cells are allowed to divide. So that G1 checkpoint is relying on some key proteins to do their job. And remember, those proteins are called tumor suppressor proteins. And the reason they're called tumor suppressor proteins is if they're working correctly, they're going to kick bad cells out of the cell cycle and not allow them to reproduce. If you inherit a bad version of one of the genes that code for your tumor suppressor proteins, you are more likely to get cancer. It doesn't mean you will 100% get cancer. It just means if you're exposed to any of those things that cause DNA mutations, you're not going to have the ability to kick those bad cells out of the cell cycle. So we know that people who have those mutations are more likely to develop cancer, but that is not the primary cause of cancer. Any questions about that? You can find out if you have a mutation in one of your tumor suppressor proteins. I personally would not want to know that, but we have the ability to find that out. Okay, and then somatic mutations can occur in any of your cells of the body except for eggs and sperm. Okay, so you don't pass those on to your offspring. So if you smoke and you end up with lung cancer, you cannot pass that on to your children. And this really shows you that inherited forms of gene mutations that cause cancer are the minority, really, cause of cancer. Most cancer is caused by just mutations in the cells during your lifetime. This both means you got the bad tumor suppressor protein and you were exposed to something. Any questions about that? I think it's just interesting to see that. Sorry, I haven't checked the chat for a while. Let me just check really fast. Oh, we're good. Okay, so this is really just talking about what cancer is. And cancer is a disease in which abnormal cells grow out of control because they don't follow the normal rules of cell division. In fact, if you were to look at the cell cycle for a cancer cell, the clock is very different. They're constantly dividing. And I think I draw this in the video where, you know, really G1, S, and G2 are pretty small, and cancer cells are dividing and dividing and dividing and dividing. They're spending most of their time dividing. And a lot of chemotherapy drugs target all cells at metaphase of mitosis. And the premise is cancer cells are constantly dividing. If we just keep killing all cells that are at metaphase of mitosis, we're going to hit a lot of cancer cells. And of course, you know, you hit all these cells that are rapidly dividing, your skin cells, your stomach lining, which is what causes, that's the main cause of nausea in patients going through chemotherapy, hair loss, fingernails, anything rapidly dividing is going to be affected. So there are signals that govern how normal cells divide and really, dictate that normal cell cycle. This is the abnormal cell cycle. But cancer cells really become immune to those signals. And we're going to talk a little bit more about that. Those stop signals and start signals in the cell cycle don't affect cancer cells. So this is just kind of a list of some of the external and internal factors that can cause cancer. So obviously we know a lot of these external factors are pretty well documented. Look at the chat. Am I just not seeing all the chat? I keep seeing things pop up, but that's weird. And then factors within the cell, we've just briefly talked about inherited mutations, but also you can have changes in different hormones within your body that make you more likely to get cancer. And hormones, by the way, hormones aren't just estrogen and testosterone. I mean, hormones include a lot of different chemical signalers in the body. And you'll learn all about those in physiology. Different immune conditions that make you more likely to get cancer, because for the most part, if you have a healthy functioning immune system that is picking out cancer cells, you have natural killer cells as part of your immune arsenal that will destroy cancer cells. So you have T cells in your bloodstream that are programmed to destroy cancer cells. So if you have a certain immune condition, those cells are flying under the radar of your immune system and they are allowed to divide. And really a lot of cancer therapy today is focusing on that, taking advantage of the fact that we can pull out your own white blood cells and we can reprogram them to fight the cancer cells. Not in all cases, but that's part of what some types of cancer are using that type of treatment today. That's a great question, Rachel. So Rachel's asking that if drinking alkaline water can affect cancer cells. Yes, I've heard that before. And here's what happens when you drink alkaline water, it goes directly to your stomach, where it mixes with stomach acid and becomes pH 1 to 2. So there's no possible way. And also, even if you put yourself on an IV and pump that alkaline water directly into your bloodstream, the carbonic acid buffer system, along with other buffers in your blood, are going to change the pH of that water immediately if it doesn't kill you. I mean, it would probably kill you if you put it straight into your bloodstream. Because remember, enzymes and other proteins can only function in a narrow range of pH. And if you start messing with the pH of your body, it's going to have really negative ramifications. So we have a lot of things in place to prevent that from happening. So anyone who's selling you alkaline water is just from you. Because again, it really just becomes acidic the second it hits your stomach. Water is one of the only things that is absorbed directly through the stomach lining. It doesn't have to get to the small intestine before it's absorbed, but first thing that happens is it hits the acid in your stomach and it turns to acid. So there you go. That's a great question. I'm often asked that question because I know a lot of students who drink that alkaline water. And I will also tell you that when my stepson was in the hospital, one of the things that people would bring him on a regular basis, like people from the outside who would come to visit him, is they would bring him the alkaline water. And we would just laugh and throw it in the trash when they left. They meant well. And then, of course, somatic mutations are those ones that are not inherited. Okay, so what really causes the cancer, though, is not just that the cells are dividing out of control, it's that they keep growing out of control and they cause tumors. Tumor is just a mass of abnormal cells. And what happens is they start really affecting normal tissue, but as you're going to see in a minute, one of the main things that tumors do is they attract blood vessels to grow to the tumor to supply the tumor. It's crazy. They release chemicals that recruit blood vessels. And so the organ that they're growing on is compromised, and the cells of the organ start to die because the tumor is getting all of the blood supply. So the tumor cells secrete chemicals that cause an increase in blood flow to the tumor at the cost of not sending enough blood to the surrounding tissue. So the normal surrounding tissue starts to die. So it's not really that the tumor is kind of, you know, taking up room and putting pressure on those normal cells. It's really, a lot of it is a blood flow issue and really compromising the function of that surrounding tissue, not allowing that normal tissue to do its job, because it is taking up room, but also because the blood flow is now going to the tumor. Normal cells have something really important to understand, and that is, and you've all experienced this, right? You cut your hand and the cells just grow until they touch each other, and then they stop growing. They don't just keep growing up into a big old lumpy mound on your skin. Now, sometimes you do get a scar where the cells don't perfectly divide to fill in that space, but you're not going to end up with this giant tumor on your skin as a result of that. And that's called density dependent inhibition, when crowded cells stop dividing. So they start dividing and they only form a single layer, and when they touch each other, then they stop dividing. If you scrape away some cells, they're going to grow and come together, but they're not going to keep growing. Cancer cells don't do that. Okay, cancer cells do something very different. Okay, they don't have density dependent inhibition. They just keep growing, and that's how we end up with a tumor. So rather than just forming a single layer, they can form many, many layers, and it's not even in a regular pattern. They can go on dividing indefinitely. And you've probably all heard stories of, there are a few cell lines in labs, and those cell lines have been around for many, many, many years, and they just keep growing forever. They never die. The cells are immortal. The normal cell cycle controls that would cause eventual cell death aren't there. Also, normal cells have anchorage dependence. They must be anchored to some sort of substrate on the bottom of the tissue layer, but cancer cells can just break off and travel somewhere else and grow. Okay, so let's think of it this way. Let's think of maybe your kidneys. If a kidney cell breaks away from the kidneys, it's not going to travel somewhere else and start growing a kidney on your liver, right? That's not even possible. Our cells stay where they are. They have to be anchored to a substrate. But cancer cells are completely free of that requirement. They can travel anywhere in the body and start growing, which is really scary. These are some of the other characteristics of cancer cells. I already said they're immortal. They produce go signals of their own. Okay, so they can produce a signal that causes the cell cycle to accelerate, and also cause growth factors to be recruited to cause them to grow and develop even faster. We're going to talk about what oncogenes are in a second. They override stop signals from the tumor suppressor genes. So we have oncogenes that tell the system to go, and we have tumor suppressor genes that tell the system to stop. And cancer cells really defy both of those. They're not killed by the immune system. So remember that when cell death happens, it's called apoptosis. That doesn't happen in cancer cells. This is what I just talked about. The technical term for that, which you don't need to know, is angiogenesis, but this means that they release chemicals that recruit blood vessels to the tumor and supply that tumor with a blood supply. And then also they can spread to other sites of the body. So when a cancer cell breaks off and travels and starts growing somewhere else in the body. That's metastasis. And that's where that term metastatic tumor comes from. So there are two kinds of tumors, benign tumors and metastatic tumors. Metastatic tumors are growing somewhere outside of the origin of where they started. So that's the cancer cells broke away and now a tumor is growing somewhere else in the body. Okay, so when we say cancer genes, really those fall into two categories. And again, not all cancer falls in this category, but when you inherit a bad gene, it's really one of these two kinds. So you either got a bad copy of the tumor suppressor gene or an oncogene. And to date, we know of approximately 400 different somatic cancer genes. There are definitely more than that out there. So tumor suppressor genes. Okay, so a normal tumor suppressor gene, remember G1 checkpoint, this R really just stands for regulation. That's when the cell decides whether it's going to continue or if the cell cycle is going to be stopped. And tumor suppressor genes would normally prevent a bad cell from growing and dividing. If you have a mistake in a tumor suppressor gene, it's going to allow cancer to grow. And this kind of shows that. So normal genes prevent cancer, but if you have damage to those genes, it's going to lead to cancer. And again, the main one I talk about in the video is the P53 protein. So we would be talking about the gene that codes for the P53 protein. We're moving toward talking about heredity, and it's important to realize that for every one of your genes, you have two copies. So you've got a copy from the egg that made you and a copy from the sperm that made you. So if this is your tumor suppressor gene, your P53 gene that codes for that P53 protein, two good working copies of that gene, you have a really good cancer control system. Okay, you're going to get some abnormal cell growth. And for the most part, if everything else is going well, these two healthy functioning genes are going to make proteins that are going to suppress tumors, keep you from getting tumors. If you have one bad copy of the gene and one good copy of the gene, you're still making good tumor suppressor protein. So you're still pretty unlikely to get cancer. The problem is if you have bad and bad. Now you don't have a normal copy of that gene and you don't have the ability to make a normal tumor suppressor protein. Now you're exposed to an environmental toxin that causes a somatic mutation. You don't have the ability to kick that cell out of the cell cycle. And that bad cell is going to reproduce outside the controls of the cell cycle. And that's how you end up getting cancer. This is only one way. This is only one of the mutations that you can get. But that's a pretty common one. The other type of mutation you can get is you can get an oncogene mutation. Okay, so these are genes that promote cell division in a controlled manner. And if something goes wrong with one of those, then you're going to get cancer. Okay, so these would be genes that say you have an injury and you need to do some repair. These oncogenes are going to promote cell division to repair those cells. But again, if that goes bad, you're going to have accelerated cell growth and cell division, and that results in cancer. You'll learn about oncogenes in microbiology and I believe also in physiology. By the way, while I'm saying microbiology and physiology, a couple of you have come to me and asked for advice about what to take next. And if you have questions about individual instructors or what class you should be taking next, be sure to ask. I normally highly recommend to students that they should take microbiology next. I know that's not what the counselors tell you, but that's what we as biology faculty believe results in the most success for our students, because you have to remember all of this stuff for micro to be successful. Otherwise, you have to relearn all of this at the same time you're trying to learn the new material in micro. So we found that students are most successful when they take micro next. Having said that, micro requires you to be in person for the first 8 weeks of lab. So if you can't do that, either because of a job or just not wanting to possibly get exposed to COVID, then this will be the one and only time I tell students, then don't take micro next semester. Try to take physiology instead. Anatomy can be taken anytime. It does not have to be taken before physiology. Physiology is all cellular processes. You don't need to know where your liver is, all the different bones of your body, every tubercle and foramina of every bone. You don't need to know that to take physiology. They're not connected. So you can take physio before you take anatomy. So if you have questions about that, be sure to ask me. Okay, this is just quickly some cancer stats. This is astronomical. In 2020, 1.8 million new cases of cancer will be diagnosed in the US. And 606,000 people will die from cancer. That's pretty crazy. You can see here in order: breast cancer, lung cancer, prostate cancer, colon, melanoma. You can read the list there if you're interested. So for men, almost half of the cancers are prostate, lung, and colorectal. And for women, it's breast, lung, and colorectal. Colorectal screening for cancer has increased dramatically in the United States, and this is why. It's because it really is one of the primary cancers that still kill people. And early detection obviously is really key. So this just gives you some more stats for 2020. These are all really current. I just took them off the American Cancer Society website this morning. So this is men and women combined. But in general, mortality rate is higher in men than in women. So 100, really basically if you round that, 190 per 100,000 for men and 136 for women. And you can see that race and ethnicity do play a role in cancer rates in the United States. And this is huge. Cancer, number of cancer survivors. I mean, 10 years ago, we probably never imagined that we would reach this milestone of having that many people survive cancer. And it's, by 2030, they're estimating that it's going to, just keep going up, which is pretty awesome. So, as future healthcare providers, many of you will be dealing with cancer. And I just think it's really important to understand just a little bit about what causes it. I had one more set of slides. I'm going to stop sharing for a second so I can do a new share. Can you see this new set of slides?
Yep.
Okay, good. Some of these questions: can cancer be prevented? How is cancer diagnosed? What is the link between genes and cancer? What causes cancer? I mean, those are kind of the biggest questions that people typically ask about cancer. Obviously, we know some things we can do to prevent cancer, but a lot of it is just luck, unfortunately. We're all obviously going to eventually die. We don't know what from and how long we have on this earth. And unfortunately, many of us will get cancer. And early diagnosis is obviously key. And unfortunately, that relies on you having really good health care, which doesn't always happen. And so often now people who have cancer, still today go in with all these different health issues. And believe it or not, it's tough to diagnose, really, unless you're having a PET scan or some specific scan, which your insurance company isn't just going to send you for a $25,000 scan. They really kind of rule out everything else before they get to the cancer diagnosis in a lot of cases. So just because you go get blood work done doesn't mean they're going to catch your cancer. So that's the problem. A lot of times you just have to get really lucky and have it diagnosed early enough before it gets too far out of control. Because we know that if you get to, stage 3 or 4, you have a much less chance of survival than if you catch it at stage 1 or 2. This shows you kind of the different types of cancer. So carcinomas are one category, sarcomas, leukemias, and lymphomas. Those are the 4 major categories of cancer. And so, breast cancer, prostate cancer, colon cancer, and lung cancer, those are all carcinomas. We also have skin cancers that are carcinomas, but those are not deadly. Then there are the sarcomas, and then obviously the blood cancers and the cancers of the lymph nodes. When you take physiology and anatomy, if you don't already know this, you'll learn a lot of terms that will make you really good at Jeopardy and figuring out what different words mean, because all of these prefixes, these Latin prefixes, have a specific meaning. And so a lot of times the name of the carcinoma or the sarcoma is combined with a word that tells you where that cancer is found. You don't need to in any way memorize any of this for the test. I'm just showing you that that's where those terms come from, is that that's just what those mean. This kind of shows you just what we already learned, that a normal cell, if it has an error, it's going to get kicked out of the cell cycle. And it will either be stimulated to commit cell suicide, which means, again, all the lysosomes open up and digest that cell, or one of your white blood cells is going to destroy that cell. But in cancer, that obviously doesn't happen. And bad cells are allowed to divide and reproduce. This picture shows you not only recruitment of blood vessels, but it shows you metastasis, that those cancer cells can break away and travel somewhere else and invade new tissue if left untreated for long enough. That's kind of that's when it becomes deadly, is when that happens. So what is the difference between a benign tumor and a malignant tumor? A malignant tumor is invading other tissue and has recruited blood vessels, and you can see that here. Versus a benign tumor, it's just local, it hasn't spread. So they generally say that's not cancer because that growth is still controlled. Okay, so you have some abnormal cell growth happening there. You can usually just have it removed with minimal damage, and you don't have to worry that it has spread somewhere else in the body. So as you get old, you'll have stuff that accumulates, especially on your skin. And most of that is not, fortunately, most of that is not cancer. Let me check the questions again, sorry. Any questions about any of that? This is the tough part, like I was saying, early cancer usually doesn't have symptoms. So there are various levels of cancer screening, cervical cancer, prostate cancer, breast cancer, colorectal cancer. Those all have specific tests that you can have done to detect those cancers early before you would even possibly start having symptoms. So this just shows you some of those cancer screening tests. And of course, we now know that most cervical cancer is caused by a virus, the human papillomavirus, HPV. And now kids are vaccinated for that. So hopefully cervical cancer rates will start going down dramatically. I mean, how crazy that we now have a vaccine for a specific type of cancer. That's pretty awesome. Obviously, mammogram is first line in detecting breast cancer. Prostate and ovarian cancer screening, there's a blood test that's done for that. And then colorectal screening, the most common test that they do is the fecal test, where you just mail in a sample. And then the more diagnostic test is the colonoscopy, which everyone is supposed to get anywhere between 50 to 60 for your baseline, depending on your overall health and diet. If you eat a lot of fiber and have a really healthy diet and no family history, you can sometimes wait till 60 to get that done. But most doctors recommend it earlier. So in the biopsy, they're looking at the cells, okay, and they're looking at a few different things. They're looking at the genome now. That's pretty new and pretty awesome that they can actually look at the genome of those cells and determine whether they're cancer cells or not. This is looking at the different proteins that are produced by those cells and are those proteins normal or not. And then just looking at the pathology, looking at the slide to determine whether those cells are dividing and growing normally or not. So these are just some of the things that a pathologist would look at for a normal versus a cancer cell. So you can just kind of see the difference there. You don't have to memorize any of that. Just kind of wanted to show you that slide because I think it's interesting. Like, okay, well, how can they tell that you have cancer by looking at your tissue? Well, the cells just really are completely organized differently. They don't really have the normal cell shape. And, you know, for my stepson's leukemia, he actually just was having some intestinal issues, and we took him in and they did some blood work and they figured out that he had C. diff. Well, he'd been on an antibiotic for his skin. So we're like, wow, that's really weird that he ended up with C. diff. Well, it was because his immune system was really compromised, and they did just some basic blood work and realized he was pumping out millions of immature, malformed B cells. So if he didn't get the C. diff from the acne medication, it would not have been diagnosed. It was just luck. Again, a lot of it is luck in catching something early. So this is just showing, again, you know, the difference in cancer cells when you're looking at tissue under the microscope. This is just showing you that, you know, if you have, this is what we call in the US stage 1 or stage 2, your survival rate is much higher than if you have stage 3 or stage 4. And what that's looking at is the degree of tumor growth and how far that has moved through the body. Has it metastasized somewhere else or not? And you can look at this is just for melanoma, stage 1, 2, and 3. This shows 5-year survival rate. As far as what causes cancer, we've already talked about that, but I thought this was really interesting. You know, this is one of those questions of, is it genes, your behavior, or the environment that's causing this difference? Because if you look at the rate of colon cancer in Japan, it's pretty low. So that would indicate that when they move to the US and it suddenly goes up, that has to be environment or behavior, right? Because if genetically, as a population, they don't have a high rate of that, but maybe it's because they have a healthier diet, right, in Japan, versus US citizens, much higher. So these kinds of studies are really interesting in trying to figure out the links between some of these cancers and our behaviors and environment. And then look, stomach cancer, that's crazy. It's much lower in the US than it is in Japan. This is just another list of different, this is just the carcinogenic chemicals in tobacco smoke. I mean, that's a lot of different things in tobacco smoke that causes cancer. A lot of stuff. This just shows you cigarette consumption and lung cancer rates and how they've gone up over time. By the way, this is starting to go down. If you look on the American Cancer Society website, you know, this is really starting to go down as fewer people are smoking now. So that was smoking. This is UV radiation. So looking at different regions of the country. Again, this is not the deadly form of cancer, which is melanoma. Melanoma is not related to sun exposure. So this would be basal and squamous cell carcinoma that they're talking about. So the sunnier areas with higher UV radiation definitely have a higher rates of those kinds of cancer. And radiation, X-rays. You know that they used to, when your kids would go in to get new shoes, they had to go to a shoe store to get them. And they would have them put the shoes on and then they would X-ray to see if the shoes were fitting. And sometimes they would do it like 20 times in one visit to the shoe store and nobody was wearing a vest. You know, they didn't know that it was bad. They're like, hey, cool, look, we can see your foot bones and how they're fitting in the shoe. And of course, we now know that there are some viruses that can cause cancer. And HPV is a good example of that. The virus changes your DNA enough that your cells start growing abnormally. That's pretty crazy.